Hello everyone, and thank you for joining us for today's webinar. I'm sure by now we've all either been dreading being hit by the often referenced tsunami of data coming our way, and many of you might be finding that to be exceptionally challenging as we speak. So we are very fortunate to have 4 speakers to not only explain how to over those challenges, but also how to turn them into opportunities. If you have any questions at any point, type them in and we'll address them as best as we can at the end. If we don't get around to your questions, we'll try to get your response via email. So without any further delay, I'd like to introduce our first speaker of the session. First up is Edward Farmer, who is Executive Vice President of Communications and New Ventures at WuXi NextCODE. Most of you might know him for his work at Decode Genetics, where he helped to build their profile as a global leader in human genetics, overseeing media and investor relations and launching major websites on gene discovery, DNA-based giant diagnostics, and drug discovery and development. Over to you, Edward.
Thank you very much. I'm not sure whether you can see my, let's see, there we go. Yeah, thanks very much, and thanks Rafael and Informatics for the invitation. It's a pleasure to be here. I just wanted to take a couple of minutes before handing you over to Hákon Guðbjartsson, who's the person you're really going to want to hear from. Just to outline a bit about who WuXi NextCODE is, what the challenges are that we see in the space in terms of big data, and how we're addressing those in very general terms. Hákon will then take that up in technical terms. So in the most basic sense, what WuXi NextCODE is doing is really providing a global platform for the use of genomic data at scale and across different projects and countries. I'm sure most of us on the call today are involved in some degree, in precision medicine. I would define that very simply as being the use of the genome to improve health and healthcare. And really, the revolution of the past few years, as we all know, has been the ability to economically and easily sequence whole genomes. Our field used to be defined by data poverty, really. We were trying to extract information from poor data sets, and now we're able to do so. We're able to sequence whole genomes with ease, and so now we're trying to extract novel information from very large data sets. What's coming out of that, of course, is both better diagnostics. I think rare diseases and classification of cancer are 2 of the things that are leading the way there. More targeted drugs, better targeted drugs, and more efficient development. Clinical research has been greatly helped by genomics, and then also personal wellness. Those are the sort of 3 main applications that that we see in the field and that are being rolled out worldwide. As we're able to sequence genomes, of course, every person starts to represent a large quantity of digital data, right? So we're talking about somewhere between 100 and 150 gigabytes of data per person, and the genome is in essence starting to define big data itself. Right now, probably the biggest source of data is I think cute animal videos. But in the next few years, the genome is going to dwarf that as a source of big data. And so the challenge really is how do you wrangle that data and use it and turn it into meaningful insights for the application that you're trying to address. A bit about WuXi NextCODE. So our position in this field is that we're a spin out of Decode Genetics in Iceland. And Decode is the first and still by far the largest population genomics effort in the world. Been wildly productive in discovering new links between the genome and disease. And the reason that we're here today is that one of the things that we needed to do when we were at Decode was that we sort of confronted the big data challenge before anybody else. Hákon will tell you that he's not any smarter than anybody else in the IT field. We can disagree with that or agree with it, depending. But the fact is that he and his team confronted the challenge of having very dense SNP data on hundreds of thousands of people, and the traditional databases were proving not up to the task of storing and querying that data efficiently. So we spun out of Decode in 2013. We set up our sort of business and genetics, and now AI and deep learning headquarters in Kendall Square. Our IT group is still in Reykjavik. And then in 2015, we merged with WuXi AppTec's Genome Center in Shanghai, which gave us both a state-of-the-art sequencing facility, CLIA NCAP certified, as well as a foothold in the China market, so that we're really able to span the globe and applications in a way that's not been matched yet. What that gives us the ability to do, of course, is everything from identifying cohorts all the way through really novel applications of deep learning to the genome, which is essentially, it's very interesting because you're going from biology, you're digitizing biology, and in fact, through AI, you're able to go back to biology. But what we'd like to focus on today is very much the middle portion of this span of digitizing, managing, and analyzing genomes, and the management is, I think, going to be is our secret sauce, and is going to be the core of what we're going to talk about today. And we're very happy to be in touch with the people at Informatics because we're able to apply and have been called upon to apply our technology across a range of applications. So we're developing both services and our own solutions, for example, in China, across the whole span of precision medicine. That includes basically all of the big projects at the population scale, precision medicine applications where we're looking at many people, as well as clinical and wellness applications where you're essentially dealing with an N of 1 and trying to either diagnose the disease or help an individual to optimize their own health. With that, I will turn things over to Hákon to explain a bit more about the GOR database that is kind of at the heart of what we do and its application. So Hákon, over to you.
Thank you, Ed. I'm very happy to be here in this webinar and being able to describe our technology for mining and organizing and sharing genomic data. But before I go any further, I would like to sort of emphasize that sort of very much of the drive behind our design and of our architecture is the recognition of the fact that whole genome analysis and whole exome analysis is making the overlap of clinical work and research much bigger. And this is partly due to the fact that, unlike with panel-based approaches and array-based technologies, we are discovering new variants as we measure patients. And also the fact that this data is so comprehensive, that data that you capture for clinical purpose makes actually ideal data for leveraging in research down the road. So this brings makes the overlap between clinical work and research work much bigger than ever before. And we think that we have an extremely comprehensive platform to serve both of these needs, both the clinical work and also the research aspect of the clinical work. And we have what we call the NextCODE platform, which addresses not only highly scalable storage, upload of data into the cloud, but also elastic compute for batch jobs that are based on secondary analysis, which are extremely computationally intensive, but also elastically scalable database to support user-driven queries and analysis, both for clinical work through our clinical sequence analyzer and also in our sequence miner research workbench application. But I'm going to focus on the the database technology that is part of our platform. And while we were at DCODE, we recognized what was happening as more and more people were starting to work with SNP-based data, where you had millions of data points that people stopped or running basically into difficulties using conventional relational database technologies for SNP-based data, where you have million data points per individual. And with the advent of high-throughput sequencing, this became even much more difficult than before. And so what we have been seeing in the bioinformatics space is what I refer to as the exodus from conventional relational database systems that actually were made very popular in the 70s and revolutionized the business world in terms of data management. And when we embarked on this heavy, large-scale sequencing project while at DCODE, we thought we would like to actually reinstate some of the good database practices that came across with the relational database systems. But for making that possible, we needed to implement them in a strikingly different way. And then we came up with this technology that we refer to as the GOR system or the GOR database. And what that is in short is essentially a relational database system for carrying out analysis on genomic data on large scales, similar to what you would use the conventional relational database systems for conventional business data that is typically orders of magnitude smaller. This technology is described in great details in a publication that we made available in Bioinformatics last summer. But in a sense, it's a database technology founded on the principle of streaming data and merging data from many different data sources. And so the key thing is not try to shuffle data or use index technologies, because they create a huge burden on data updates. And so the idea is to stream data as much as possible and basically analyze the data based on the genomic position, which is sort of in heart the most natural organization of data for most of the use cases that we observe in genomic analysis. And so that's what we mean by the GORE architecture. So it's both storage of data, also building algorithms that leverage this principle of streaming the data in a consistent order. But it's also very important in the relational database world to use a query language, and the sort of the de facto query language in the conventional databases is a SQL query language. We, however, decided actually not to use SQL. First of all, SQL is not being used very much for genomic data today because of this exodus from the database systems, but also there's a lot of legacy and sort of legacy constraints that we would be, stuck with. So we decided rather to merge the sort of the Unix common syntax together with some of the key features of SQL, and we came up with what we refer to as the GORE pipe syntax. And in essence, it's very similar to the sort of a Linux shell pipe syntax that many bioinformaticians are familiar with. But where it's strikingly different is that you can also make highly declarative queries, and so-called nested queries, like people observe in SQL language, are seekable, meaning that you don't need to materialize as much data. And this also then boils down to much more flexibility in developing queries, but also much more efficient execution, because you don't need to store the data. And so I lost my slide here. Where am I? 50 minutes.
The slides you were.
Yeah. Okay. Yeah. So, and this database framework that we have developed is very convenient and highly compatible with so-called conventional bioinformatics pipelines, as I call them, that rely on batch processing and dealing with data such as from the conventional variation calling algorithms such as GATK and so on, but also for interactive analysis such as from query systems or genome browsers. And so one of the key things by having the data organized in a GORE fashion and having algorithms implemented that leverage that, it allows us to organize data in a normalized manner. And this is a principle very well known in the conventional database space, which basically means that you can have more flexibility for changing data. We, of course, also support denormalization of data, but having extremely efficient way of doing joins of data allows us to use the principles of normalizations like in the conventional database systems. Additionally, we support materialized views. This makes development of heavy analysis much easier for application developers and leverages caching of data, and basically means that the development effort is very much reduced for those who use this query framework. We support parallel execution, both on what we refer to as the genomic level as well as on sample level, and I will explain that in a little bit more detail. So essentially, the sort of the two extremes data layout is, if you imagine when you retrieve data from the sequencing instrument, you are essentially getting files that deliver data on a per individual level. And the left scenario, you can imagine that being like a single BAM file where you have one data on one individual or one subject lined across the genome. But when you do cohort analysis, often the trend is to do so-called joint variation calling. and try to organize data in the sort of transposed fashion, having very rapid access to all the data of multiple individuals, let's say from the same mutation or from the same gene. This is very nice when you can do it, but this pattern of trying to transpose the data is unsustained when you have very large cohorts, and data updates become increasingly difficult as numbers of samples in the database grow. What we have done is a compromise between the two. We support individual file-level organization of data, but also so-called bucketization. And this allows our database system to basically choose an access pattern based on the number of patients in the query and get a very good compromise between efficient analysis as well as cheap update dates of the data in the database system. This also allows us to leverage the organization of data for parallelization. So not only can we do parallelization along the genomic axis, but we also have special instructions to allow queries to be parallelized based on how they are organized into buckets on the file system. And I mentioned earlier that this core architecture allows us to do extremely efficient joins, and joining data along the genomic position is one of the key use cases in data analysis. And parallelization is important, but it's also important to do things efficiently, because in the end you are going to pay for all the hardware that you use for the parallelization. And as an example, our core system does genomic joins 10 times more efficiently than the Spark system that uses more conventional types of joins for general purpose business analysis. And this is just an example of a Spark query used to perform these comparisons for those who are familiar with the Spark syntax. And the last piece of code there is a small query in the Gore pipe syntax that does equivalent query to the Spark one, showing that the CPU utilization is 10 times higher in our core system. We also support addition of new commands, and this is where the pipe syntax is much more flexible than the SQL syntax. It's very easy to introduce new commands. These can be built using JVM languages such as Java or Scala, but also more importantly, also using languages like R, where there is lots of statistical libraries available, or Python, or even Bash command lines. And these can be plugged into the system dynamically by the user, or whitelisted commands set up by the database administrator. We support elastic scalability, which is extremely valuable for cloud deployments. And this scalability is not only for CPU, we are also supporting scalability on the CAS file level. And our system can read data not only from conventional POSIX file systems such as NFS or EPS volumes, but also from object storage. And we have a built-in mechanism to migrate data from these different tiered storage levels into our system for more rapid access to these core workers. And both of these levels, both the compute nodes and the CAS file nodes, have elastic scalability. And this is in a way where our architecture is very similar to the one from Informatics, where we're leveraging tiered storage. And we are actually looking forward to be able to then take this further and use the high compression utility from the Informatics solution and have that transparently under the hood of our core database system. We also support federation, meaning that we can read data from not only file system but also URLs and things like that. And finally, I would like to mention that this is sort of the underlying database engine that powers our clinical sequencing analysis platform. And the key thing there is not only just to be able to look at one sample at a time and compare how the mutations of that individual contrast with publicized mutations, but we see that the clinical picture and the work in clinic is more and more going to rely on data that gets captured into what we refer to as ongoing sort of variation collections sort of per site, where you're going to have lots of not only curated mutations by the clinicians, but also just a large collection of mutation data from patients that have well phenotyped and characterized samples. And that becomes ever more valuable information to contrast with than data taken from publications. Because with this flood of mutations from next generation sequencing technology, we believe the literature way of publishing all these mutations is not going to be sustainable. But essentially, the CSA system, or the clinical sequence analysis, is a mechanism to take you from next-generation sequencing into a clinical report in a smooth fashion, and also curate the mutations you come across in the process. But finally, we also have end-user application we refer to as the sequence miner, and essentially like a workbench for diving into the data, tapping into the GORD database, visualizing data in a genome browser from queries in the GORD database, or for posing queries directly into the system, setting up canned queries in report builders and so on. So this sort of concludes my overview of our applications. Thank you.
Thank you, Hákon. Next up presenting is Eran Segal, who is a professor of computer science and applied mathematics at the Weizmann Institute of Science. His research focuses on nutrition, genetics, microbiome, and gene regulation, and their effect on health and disease. His aim is to develop personalized nutrition and personalized medicine. Over to you, Eran.
Okay. Hi, can you, am I on? Can you hear? Okay.
Yeah, you are on.
Okay, excellent. Okay, so yeah, hi everybody, and thank you for the opportunity to present. So I will focus on describing a little bit our research, and also the heavy use of large-scale genomic data that we are working on, and really I will emphasize from the beginning that with the large data sets that we've been using, and the more rather limited resources in academia, the collaboration with of my lab with Informatics has really been an enabler, as you will see in some of the next slides, on being able to even store and rapidly access the data that the vast amounts of data that we collect in these various studies. So my group for the past 5 or 6 years has been focusing on the human gut microbiome and its relationship to health and disease, which has been demonstrated in multiple different studies, both by us and by many others, and it's really been linked to a wide variety of different health conditions, which you can see on these slides. Our personal focus in the lab has been a lot centered on metabolic disease because of the huge rise that we've been seeing both in obesity worldwide and also in diabetes, and the clear role that our gut microbiome and our nutrition plays in the development of these diseases. And we've been involved in multiple different projects. So I want to touch upon a few. I'll mention a couple very briefly, and then spend a few more minutes on one of them. So in one study, we focused on artificial sweeteners, and which the vast majority of us consume on a daily basis, and are even recommended by the American Diabetes Association for diabetics as a replacement for sugary drinks and sugar in general. And because you typically see overweight people consuming these drinks, we thought that a legitimate question is what comes first, and perhaps the sweeteners themselves are actually the ones that may be contributing to these conditions. And indeed, in a series of many different experiments that I don't have time to go into, but have been published in the paper that you see here below, and you can access from my lab website, we've demonstrated that if you take gut bacteria and you grow them in the presence of artificial sweeteners, and you then transplant them into naive mice that never consumed artificial sweeteners, you can induce glucose intolerance in the recipient germ-free mice. And in preliminary experiments in human, we also showed that even humans who do not normally consume artificial sweeteners, when they do even for a week, that already is sufficient to alter the gut bacteria composition such that when transferred into recipient germ-free mice also can induce glucose intolerance. In other projects, we've shown, for example, that even though our gut bacteria very much live in the dark of our gut and are not exposed to light, nevertheless, they actually exhibit diurnal rhythmicity, so they go up and down in abundance during the day. And that diurnal rhythmicity is really very important for us, the host, because that directly affects and alters gene expression patterns, epigenetic patterns, metabolite patterns that are flowing around in our system, and also gene activity in distant organs such as the liver, and we've shown that that's important for even processes like gene detoxification of various drugs. In another line of work that we've invested a lot in, and that I'll spend a few more minutes on, is one where we've explored the relationship between nutrition and gut bacteria. You may be very aware of this from just popular reading, that over the past several decades, we've been really significantly changing the overall nutritional recommendations that we've been giving the public, sometimes saying, for example, that cholesterol is good and sometimes that cholesterol is bad. And so really in this project, we wanted to take an unbiased scientific approach to the problem of nutrition. And the first thing that we asked ourselves is what in our nutrition should we really be focusing on? And after a lot of thought, we've decided to focus on the postprandial glucose response or post-meal glucose response. These are specifically the changes in blood glucose levels that occur in a person after eating a meal in the 2 hours after eating a meal. And so after you eat a meal, the carbohydrates in the meal get digested and then into the bloodstream so that they can be absorbed by the different cells for use as energy. But in response to that, because the body really needs to maintain glucose homeostasis, the body secretes insulin, whose job is to reduce these levels, but sometimes it can overshoot if we have a very significant spike in glucose levels, and then glucose levels can go below baseline and causing us to feel hungry and even eat more. And not only that, but high post-meal glucose levels have been linked to a variety of different conditions, to diabetes. Obviously, this is also how diabetes is defined, but also to obesity, to other metabolic disease, to risk of cardiovascular disease, and even to cancer and neurodegenerative diseases. So we really feel that it's a very clinically important variable to measure. And with technology that has been developed around diabetes, we can also measure it at very large scale and obtain very large data sets. And that's exactly what we did. So we initiated what we called the Personalized Nutrition Project, in which we recruited 1,000 participants, each of them connected to a glucose monitor for 1 full week, obtaining about 2,000 glucose measurements per person, 2 million across the entire study. And during that week, participants logged everything that they eat on a mobile app that we developed, giving us 50,000 meals that have been logged with full nutritional values. And we were interested in understanding the relationship between these changes in blood glucose levels after meals and various components within a person. And so we also comprehensively profiled participants. So we collected body measures, anthropometrics, blood tests, questionnaires. But by far the largest amount of data in terms of quantity was gut bacteria, which we sequenced in 2 different ways, both on 16S and on metagenomics, and we've also recently genotyped this entire cohort. And so this really creates a massive amount of data sets with tens of giga of storage per participant. And so in the next slide, you can see accumulation of data in this project across time. So just the cohort that we generated from these 1,000 participants created over 70 terabytes of raw sequencing data, which we need for processing. At the time of doing this, the storage that that my lab had access to was around 40 or 50 terabytes of data, so we couldn't even store this data, yet let alone all the derived data file analyses that we have to go through from the raw sequencing data. And so here the Informatics platform, which is integrated in my lab, was really an enabler of the study, because it was able to reduce our footprint from over 70 terabytes of data to below 7 terabytes of data, which was manageable and able to store on our servers. In addition, we also wanted to compare all of our findings and the gut bacteria composition of our cohort to other publicly available cohorts. So we downloaded those. Those two were in excess of size of what we had in our cells. And so by the Genomics platform, we were able to also reduce the footprint of the publicly available data sets also by a factor of close to 15x, and also get it to single-digit terabytes of data and really allow us to perform the analyses. So the first question we were interested in is what is the overall response of different people to the same food? And so to test that, we gave participants in the morning after the night fast either bread with butter, or glucose. And really the key result, and now this was tested on an unprecedented scale that was never done before. We've demonstrated that when the same participant, which you can see on this graph anecdotally for bread, 4 participants, when the same participant consumes the same meal on 2 different days, the glucose response is very similar. But when different participants eat exactly the same meal that we provided, the response is hugely different. And you can see here some participants responding very highly, others having a mild response, and yet others having essentially no change in glucose levels after eating 4 slices of bread that we gave here. And so this really was quite a big surprise and shock, and one of the key results have come out of the study, because it really tells you that if you want a diet that normalizes and maintains healthy blood sugar levels, it cannot be the same diet for everybody. So in other words, general nutritional recommendations will never be efficient for the entire population, and nutrition really needs to be personalized if we want to maintain that. And as I mentioned, it's important not just for diabetes, but also for weight management and prevention of many other diseases. And so the next step in our research was to link this variability in people to all the various clinical data and personalized microbiome data that we collected. And we were able to develop a machine learning algorithm that improved very dramatically on state-of-the-art prediction, which you see on the left, using the amount of carbohydrates in the meal to, so going from about 15% of the variability that one can explain using carbohydrates to close to 50% of the variability that could be explained by our machine learning algorithm, taking gut bacteria and other clinical information into account. And so with that, we also went to a final step in this project and asked whether we can use the algorithm to design personalized diets for people that would maintain and improve glucose levels. And so we recruited 26 new participants on top of the previously 1,000 people profiled. We designed, using our algorithm, 2 diets for each participant. By design, the diets had to be identical in calories, but in one diet, we asked the algorithm to design meals that would be good for that participant, namely not elevating blood glucose levels, and in the others, meals that would spike glucose levels. We call these the good and the bad diet. It was a crossover, randomized, double-blinded, randomized study. To show you kind of a feel for the diet, here they are for one participant, and you can kind of take a moment and see if you can guess for yourself which one is the good diet and which one was the bad diet. I can tell you when we did this with people, our students who designed the algorithm, they couldn't guess because it's really also personalized. So some foods that you will see here were given to some participants on their good diet by the algorithm, but to others on the bad diet. And so in this particular case, it's the diet on the right, the one with the ice cream that was predicted by the algorithm to be the good diet, and the one with the chocolate was predicted to be the bad diet. And what I'll show you next are the glucose levels that actually occurred in this participant following the good and the bad diet each for 1 week. The bad diet is going to be the full trace of blood glucose levels across a week, and that you'll see that in red, and in green it'll be the trace of glucose over an entire week on the good diet. And so this is the slide, and this was really perhaps the key, most striking result to have come out of the study. So this particular participant is pre-diabetic, which you can appreciate by the high glucose levels in red and the really abnormal spikes. And then on this exact same participant, following a diet which has exactly the same amount of calories, the one I showed you before with the ice cream, this exact same pre-diabetic participant achieving fully normal glucose levels in green, which you can see here when going on the good diet. And we obtained, and it's all summarized in the paper that appears below, we obtained similar results for most participants for which we designed these personalized diets. And so if I just summarize this part, this main project of personalized nutrition that I talked about demonstrated high variability across different people in how their blood sugar levels respond to even identical meals by taking a vast amount of data. At this point, the genomic data we heavily relied on was gut microbiome. We were able to design a machine learning algorithm that predicts personalized glucose responses and then use those predictions to design personalized diets for people that improve and maintain healthy blood glucose levels. And so in closing, I'll just mention other work that we just published a couple of months ago on relapsing obesity. So this is the phenomena by which what's called the yo-yo effect of diet, by which you lose weight, but then with time you regain the weight back, and over time you gain more and more weight. We've also shown that that too is linked to gut bacteria composition, again, collecting a large amount of data, in this case from data from mice primarily, and also developing a machine learning algorithm to predict from microbiome levels at baseline how individual mice will increase their weight on the same diet based on their history of relapsing or having a history of obesity or not. So the last slide is acknowledgments to really the large group of students and researchers, both from my group and the group of Eran Segal from the Weizmann Institute, additional collaborators that we had on this project. And I'll mention again that really an enabling technology for the project was the Informatics platform, on which we stored and accessed all of our data and would not have been able to otherwise, with the resources that we had, be able to store and manage and handle this large amount of data. So with that, I'll close and pass it on now.
Thank you, Eran. Our final presenter is Rafael Feitelberg, who is the CEO of Informatics. Before joining Informatics, Rafael was Senior Director of Go-to-Market and Strategic Marketing at SanDisk. Earlier in his career, he was co-founder and CEO of Jillian Technologies, a leading provider of web application security, which is now part of TrustWave. Over to you, Rafael.
Thank you, and I'd really like to thank all of our distinguished speakers. We appreciate this very, very much, and it's a pleasure and honor to be able to be speaking with you guys today. A bit about Informatics. So Informatics really specializes in the IT infrastructure as part of the genomic process. As both Hákon and Professor Segal mentioned, in order to enable all the advanced processes and analysis in both precision medicine, in microbiome, and in others, it's really the IT part which is a critical enabler there. Not to diminish any of the other parts, but that's really the area that we're focusing on. And the area that we are able to add value in is in being able to really scale and simplify the whole processes of storing, transferring, and archiving the data, which in many cases can become a burdening bottleneck. We reduce the footprint by 60 to 90%. We do this transparently, and I'll show in a moment a bit more on how we do this at a deeper level. We've been lucky enough to be able to work with some of the leading organizations worldwide to be able to help them in their analysis in their research, and I'll share some of the use cases also of how we're doing that. One of the first things, though, to mention is that it's not just about solving or building the infrastructure for the challenges that organizations have today, but it's really a lot about building an infrastructure that is able to scale out, that is able to scale up with the huge amounts of data which are expected to come in the future. And here, this really depicts part of what I think is a critical thing to bear in mind, which is thinking that the storage race is something that one can win in genomics is very risky. Even if you assume that the amount of storage and volume of data that you're going to have increases only by 60% every year, and I have to say this is very, very, very conservative. Most market analysts talk about more than double than that. But even in that conservative case, and even in the cases where storage costs are going down by 20%, which is stronger than actually what you're seeing in Moore's law currently with the most advanced storage solutions, you still have a big deficit. And what this means is basically if you take every $100,000 that you're using for a budget on storage and IT, if you just take 3 years out with these conservative numbers, it's going to be more than triple in 3 years. And this really brings back the issue of building an infrastructure that enables organizations not only to solve the issues that they have today in transfer, storage, and archiving of data, but building really large-scale solutions that will be able to be also a solution for what are the requirements in the coming years. And this is where we come in. Being able to reduce a genomic footprint by 60 to 90% is an enabler to scale up. But one of the most important things is to do that in a way which is transparent. And what we mean by transparent is several issues. The number one is to have it in a way which is transparent, which is perfectly lossless. You'll see different solutions out there which are reducing the amount of footprint that you have, but it's very, very, very important to make sure that these are completely, perfectly lossless. And there are different algorithms out there that will do that. What we do is bit-to-bit perfect, mathematically exactly the same, and there is no loss of data. We're not taking any judgment of what data is more important or less important. We are reproducing the exact mathematical original data. This also means that there are no changes to applications. Whatever bioinformatic applications that are used to analyze the data, to query it, etc., do not need to be adapted to any new sort of data file, etc. It's exactly the same. It's totally transparent. So all the bioinformaticians can continue working the same way that they did before. The other thing is you continue using the existing storage infrastructure and scale up with it. We're not providing an alternative to the storage system. We're providing the ability to use the same storage system, but in a more efficient way. So it is transparent also on the storage system. And the last thing, which is very important, is it does not become an additional burden on your infrastructure, on your compute infrastructure, on your storage infrastructure, or cloud infrastructure. We scale up with it totally in a transparent way. So the ability to reduce the footprint, but do that in a way that is totally transparent, is the critical factor here and is what enables us to work and partner with organizations so that they can really benefit from the reduction in footprint, but focus on the science and on the important precision medicine work which is being done. In order to do that, we have a set of solutions. We focus on being able to cater to the challenges that are provided to us wherever they are, if they are on the cloud, if they are on premises, and we also have a modular utility which allows us a full flexibility of providing value whatever the workflow is. So I'll go very quickly over the different solutions. If we look at the modular utility, this is basically an executable. It's the algorithmic engine. It provides best-in-class compression and decompression and can be really integrated into different workflows. The most common workflows we're seeing this integrated into is actually in transfer of data, where the data is compressed before it is sent and then decompressed after it is received. This can be parallelized and multi-threaded and works very, very efficiently. The advantage, again, is the ability to be very flexible on the different workflows, so it's an executable that can be inserted into different parts of the workflow. Then when we're talking about on-premises storage, then our products cater to two main architectures, either a simple network model, where an NFS is used. We would add our Genomics S as an additional mount into that architecture. It compresses all the data in the background, and then when the data is requested, it decompresses it on the fly, scales out, and sends it back to the application clusters. This enables, as I mentioned before, the full transparency. The applications on the left side are not aware that anything has changed. They ask for the information. They receive the same information they requested, but from a cost perspective, from a physical perspective, the storage required was dramatically diminished. A cluster model, this is really, we're seeing a lot of HPCs working in different compute nodes. Then we would implement what we call the Informatics H. It resides on the computer nodes, scales up with them, again, compresses the data in the background. When the data is requested, it decompresses it on the fly and provides the applications with the original uncompressed data. This allows us to scale together with the compute nodes and works at very, very high performances to be able to cater to the needs of HPC environments. When we're going to the cloud, then really we start combining both the on-premises side to the cloud side. And here, due to the fact that really sequencing is done in the physical world on-premises, it's also about making sure that you can pass and that you can transfer the data in a compressed way to the cloud and work in uploads and downloads, not on the full volume of the data, but on a compressed version. And on the cloud itself, again, we have models that cater to different workflows. On the left side, you see more of a linear network model where we put a virtual AMI in between the compressed data and the compute instances, compress in the background and decompress on the fly, or on what we call the distributed model, which allows us to have several compute instances, again, compress in the background and decompress when a query is requested. These different architectures cater to different workflows. One of the advantages that we can provide here is also making the transfer from an on-premise setup to a cloud much easier by the fact that we transfer native files to compress objects and also help from the transfer from file storage to object storage through the compression and decompression. Within these solutions, we're also able to help in tiering for cloud, enabling the more efficient storage depending on the cost and tier and and cost of the different tiers on the cloud. And really, these are mostly examples of how we can really scale up to different architectures according to the needs. This has been implemented in different organizations. Professor Segal already talked about the implementation in the microbiome study. In the Sheba Academic Center, where oncology research is being done on a variety of file types, freeing up more than 50 terabytes on this specific research. Here in this case, part of the important issue was not just saving the existing storage, but really enabling them to reallocate next year's budget, not only from storage to actual more scientific work that they were planning to do. The Garvan Institute is another organization that we're working with. In that case, it was a huge amount of both archived and ongoing storage, which the technology enables to reduce the footprint of, and really a very large-scale operation in the Garvan Institute, which we're proud to be able to partner with. So this, I hope, provides some view on how the solutions and how the architecture are actually implemented. As the costs of analyzing and sequencing are going down, then the costs of storage and archiving, especially if it's for a number of years, is taking a bigger and bigger part of the budget. And by enabling the reduction in footprint and by providing IT solutions that do that transparently, we're able to really enable a lot of these large-scale activities. The way that we typically work is once you contact us, you can try the solution, and we have a free trial. We can start with a very easy showcase of what the compression would do. And the advantage there is that you can calculate for yourself how much this would save for the organization. I can say that we've been very successful in demonstrating very strongly how this would substantially reduce the budgets in both transfer and archiving. So with that said, and thanking again our speakers, I'll send it back to the moderator to see if there are any questions.
Thank you, Rafael. Moving on to questions, we've had some questions here. I'll start with, maybe start with you, Rafael. What other alternatives are there out there for lossless compression for genomics data, and how are Informatics different?
Thank you. So, first of all, when we're talking about compressing, it's important to answer it in their two aspects. One is the compression algorithm itself, and the second is how do you make it into a scalable IT solution. Today for FASTQ, probably the golden standard is GZIP, which I think everybody's familiar with. At an algorithmic level, we would compress it around 4 times better than what ZIP does, so strong advantage over that. There are some other open source algorithms out there, but from a compression performance, we definitely have a best-in-class solution, and that's part of what we provide in our free trial, the ability to try that out. But what is even more important is how do you implement that into a scalable solution, which is a lot of what the effort of the team and the technology developed and IP is. How do you create and how do you implement that compression in a way which is totally transparent to the architecture? And in that, Informatics is pretty unique in being able to combine the two ones together.
Thank you. I think I'm just going to continue with you, Rafael, as it looks like some of these questions are pointed towards you. What trends do you see in genomics data analysis, storage, and archiving, and what do you think people will do differently in 3 to 5 years from now?
Thank you. I think this goes also very much to the presentation that we heard from WuXi and Hákon. I think there is a very strong move to the cloud. The many research which started really on local academia and on premises, in order to scale up, needs to take advantage of platforms such as the WuXi platform and be able to really use the cloud as an enabler for all of that. A lot of our technology is being developed to be able to optimize that process. That's a lot of our core competence and competitive advantage, being able to use compression in a scalable way for the cloud. But I'll also refer this to Hákon. I think what WuXi NextCODE is doing today is really building a lot of this future.
Yeah. Also a question here that says, what are the estimates of the potential revenues for the stored data? Would returns exceed costs of data generation, collection, curation, analysis, and storage?
I'm not sure I totally understand the question, the potential revenues of stored data. If the question refers to the economics of scaling up the economics of the compression, then the compression works on whatever the costs of storage is. So it really goes together. And one of the important things to bear in mind there is the archiving and storage of data over time. In many cases, due to regulation and due to clinical requirements, the data needs to be stored for a number of years, so it really compounds, and being able to store the data there over time becomes more and more important. But I'd really like also to pass the question on to Hákon and WuXi NextCODE on how they see the future of this going.
Mm-hmm. Next question is for...
Just a second. Yeah. Hákon, do you want to address a bit the issue of how do you see? Yep.
Yeah, I mean, we see storage as absolutely the biggest item of the operations of the data management. And of course, the better compression you have, the less storage footprint you have. And from what we see, the additional computational cost that you incur from using compression is relatively small compared to the actual storage footprint. So sure, yes, being able to reduce the storage footprint is extremely important. And the way I see it, we have certain sort of academic sort of standards today, like the BAM compression and the GZIP compression for FASTQ files and so on. And I think going forward, we're going to see more of these sort of advanced technologies being part of the core platform.
Mm-hmm. Once you're on it, Hákon, there is a question here saying, you mentioned clinical work on the platform. I did not see an EMR on the platform. Is there one? And if not, what is the interface to the clinical data?
Yeah, so when it comes to the EMR, it's the answer is sort of twofold. When it comes to the clinical analysis, we have built into our application mechanism to capture certain phenotype codes, such as HPO, ontology-based clinical phenotypes. But we are also moving towards tighter integration with EMR systems. They're mostly looking towards the Smart on Fire integration standard for being able to automatically retrieve clinical data and make that part of the ordering process as data goes from hospital into the clinical lab. So that's sort of on the clinical side. Then when it comes to the research aspect, we also have a special system which is actually not the core system, but more based on conventional relational database technology that allows us to basically tap our query tool that we have developed to basically interact, for instance, with conventional clinical warehouse data set. And that allows the clinical data to be kind of integrated into our sequence miner application and worked with together with genetic data in the core system.
Yeah. Another question for Informatics. How is the compression/decompression algorithm affected by processor type/performance? Does the storage require specialized disk types? And under which OS platforms does Informatics work?
Yes, thank you. So the compression and decompression does require a compute. As Hákon mentioned before, we do the analysis and really the savings from at an economical level and etc. By far are stronger on the reduction in footprint than on the additional resources. It scales up. It scales up together with the existing infrastructure, so that really, as I showed before in the architectures, you can scale up by multi-threading and or by the cluster that you're actually using. It does not require any specialized disk types. We use the exactly same storage, and we're really agnostic to what storage is being used. We're just making sure that it's taking less storage space by compressing the data, and by decompressing it at a high performance on the fly, the applications receive the same data that they were used to receiving. So we're definitely agnostic to the disk type. And from an operating system, we really work with the most of them. It's not, our technology is not dependent on the operating system, we had implementation on the different CentOS, Ubuntu, etc., etc.
Okay. I have a question here for Eran. How does the microbiome field compare with the field of human genetics in terms of data size and biological insights?
Yeah, so I would say the microbiome field is probably lagging by around 5 years, more or less, behind the field of human genetics. So there's probably hundreds of thousands of individuals or close to that have been fully sequenced. There's only thousands of individuals that have been for which metagenomics has been performed. But if we look at the trends, they are very much rising, and we see the hockey stick of data at its beginning compared to where human genetics is. So I think if you combine it all, it's very clear that there's going to be a lot of the explosion of data coming right now from human genetics is going to be followed very much by human microbiome data that is also being now generated at more and more institutions and going. I would say it's really the beginning and very early days of also industry going into sequencing microbiome, which is also going to contribute a lot to the flood of data in the field.
Yeah, it's a new question that came in right now, actually, that says microbiome versus nutrition. Did the study consider variability across the major races, say for a general vertical versus horizontal trends?
Yeah, in terms of diversity, can you ask them diversity of what was asked?
Yeah, it doesn't say. We'll just continue to the next question then, and the person that submitted the question can try and phrase it a little bit different, maybe. For WuXi NextCODE, how many whole genomes will be sequenced this next year around the world, and how quickly is this data problem growing?
I think it's pretty difficult to say with great accuracy how many genomes are going to be sequenced in the world. But if one looks at the number of sequencing machines that are out there and their capacity, we're definitely getting to the point where you're going to have hundreds of thousands and soon millions new genomes per year. So the sequencing capacity is growing rapidly. And I think that the interesting point raised by Eran just now is that DNA is one, human genomes is one piece of the genomic data puzzle, but increasingly, and we are using deep learning to attack the potential of these other data sets. We're going to need to not only sequence but also store and analyze at the same time microbiome data, RNA data, and then a lot of proteomics data as well. And as you keep building out these different data sets, all of which are growing, your computational challenges are growing sort of geometrically. So I guess you could say it is growing fast, and it is a challenge that's it's becoming more complex, not less.
Yeah. The previous question, there is a person here saying, I meant how will companies monetize the value of stored data? I think he's referring to the question that you discussed earlier. I don't know who wants to answer that, Rafael or Hákon maybe.
Yeah, I leave it to Rafael. I think it's...
Okay. So, thank you, Hákon. In general, what we're seeing is that a big part of the value of genomic data is being created by the fact that you have a critical mass of data. The more data organizations have, the more insights they're able to provide for that. If it's for precision medicine, if it is for research on like the one that Professor Segal mentioned before, if it is the population studies that Hákon and Edward mentioned before, all of these really rely on the fact that you have a huge amount of data. Just to bring back proportions of what we're talking about, a full raw sequence of a human being will take somewhere between 250 to 300 gigabytes of data. That's without backup. That's without the additional streamline of data. So being able to harness the power of critical mass of data and bring insights out of it is key. That this is how organizations are monetizing on the huge amount of genomic data. Again, through insights that are important for the development of drugs, for analysis of diagnostics, for almost anything that we're doing today in genomics, a huge amount of data is critical.
Yeah. A new question popped in here. Is there any analogous system for compressing image file data sets, or is this system unique for genomic data sets and not able to be translated to image data sets?
So I'll take this. This is Rafael. This solution is optimized for genomic data. It's reference-based. We're using actually a lot of our understanding of the data sets which are specific for genomics and is hence used only for genomic data. It's a bit like if you think about JPEG and MPEG, JPEG compressing pictures and MPEG compressing movies. Each is using different attributes which are specific for the data sets and hence are able to get more performance in compression and speed out of it. The same applies in our case. We are optimizing for genomic data and hence really both focusing specifically in that area to provide the maximum value there.
A question for you, Edward. Can you provide some examples of different applications of sequence data that require different types of access, storage, and transfer?
Yeah, we're not fully clear what's meant here, whether the different types of access to genomics data require different types of applications. Is that what's being asked for? Or?
It says if there's, if you can provide some examples of different applications of sequence data that require different types of access, storage, and transfer.
So yeah, I mean, I think you know, very many of the applications for genomic analysis have very common patterns of access to the data, and that's what we have really tried to capture in our core solution. It doesn't capture all of the use cases. There are certain use cases where that system may not be the ideal system, but in large, I think people are looking at correspondence between, you know, patients in a given segments of the genomes, things like that, or comparing, you know, several individuals across the entire genomes and finding out the differences and so on. So I think very many of these cases here commonality there. When it comes to, you know, and here I'm actually referring to data sort of post-alignment. There are, of course, also lots of different varieties and options of how you look at the raw sequence data before you do the alignment of the data. Mm-hmm. And that space is still not fully explored. I mean, I think there is you know, when it comes to retrieving small mutations, that space has not been evolving that much recently, but there are still lots of algorithms that are looking into translocations and CNVs and things like that, where there is more variety in terms of how you access the data. But I think, yeah, so I don't know whether that answers the question.
Now, there is someone here asking, will the slides be available to the attendees after the webinar is over?
Yes. Yeah, we'll make them available. Also, both WuXi NextCODE and their team and Informatics are going to be in the bio-IT coming up in a couple of weeks. We'll be glad to follow up. We're going to be there in the data storage track, actually making a luncheon presentation. I know that WuXi has a big presence there in the bio-IT show as well, and you know, we welcome you guys to reach out to us between now and then, and also to set up meetings so we can continue face-to-face during that event also.
Absolutely.
Yep, that's all we have time for today. Thank you very much for joining us, everyone, and thank you all for today's speakers. It's been thoroughly educational, and a special thank you to Informatics who made today's webinar happen. Keep checking frontlinegenomics.com to see which webinars that are coming up next month. Take care.
Thank you.
Thank you.
Thank you very much.
Thank you.
Oh, bye-bye.